Yes, thank you, Wolfram. It's a great pleasure to be in this meeting and to see you all here in Göttingen. Indeed, I changed also the title a little bit to talk about some tissue engineering muscle and how this may help to address certain aspects of regeneration in vitro. So the approach of our lab to generating human muscle in vitro is pretty straightforward. We start with human iPS cells, we make muscle cells, and then combine them with a hydrogel, mainly collagen 1, to either produce heart muscle from heart muscle cells or skeletal muscle tissue, which you can see down here. And of course, one major goal with these approaches is to recapitulate muscle physiology to the best possible way in vitro physiology and also pathophysiology. And this is in terms of morphology of those cells, but also, of course, in terms of function. This is just displayed here, so a typical twitch from a heart muscle or a typical tetanic twitch from a skeletal muscle tissue when stimulated with high frequencies. So, but what I would like to talk to you about today more is the aspect of regeneration potentially in these models and addressing some of these aspects. And we've heard already great talks about this, the efforts to try to make a non-regenerative adult heart into, let's say, a regenerative state comparable to a neonate by either differentiation of cardiomyocytes. The aspect of redifferentiation is important, the stroma is important with the fibroblast, etc., vasculature, etc. We have also become interested in one factor which I would like to show you as an example of how maybe these muscle models in vitro can also help. And this factor or this protein is DHK1. DHK1 or delta-like non-canonical notch ligand 1 is a protein that is highly expressed in development, so mainly in the fetal period. At birth, expression is low in most organs, but you typically see a re-expression of that protein in disease states or also in regenerative tissues. It's an imprinted gene. It's expressed from the paternal allele, and it shares a lot of homology to the typical, let's say, canonical notch ligands, but lacks the DSL motif. And therefore is not a classical activator of notch receptors. DHK1 is a transmembrane protein, so it has a shorter cytoplasmic part and a larger extracellular part. It also has a protease recognition site where it can be cleaved to release a soluble isoform of that protein. So it can have paracrine effects, but also endocrine effects. And there is a membrane-tethered isoform which lacks this protease site and which probably also has some different biological functions because of that. In general, it is important in development. This has been shown a long time ago by in mouse models, knockout models, where DHK1 depletion leads to growth retardation, general growth retardation, but also particular reduction of skeletal muscle mass. Yeah. And because of its role, or also known role in skeletal muscle, we first investigated in skeletal muscle models how DHK1 may behave. So we used this engineered skeletal muscle and first investigated if we can induce an injury and if after injury it would regenerate. Now, skeletal muscle obviously is a bit different to the heart in terms of regeneration because it has muscle stem cells or satellite cells that drive most of the regeneration. And first of all, we find those satellite-like cells, I would call them, in our tissues in a typical position adjacent to a myofiber underneath the laminin positive basal lamina. Those cells can be activated by, for example, cardiotoxin as a toxin, which leads to pretty much early on, like 2 days after the injury, almost complete destruction or complete destruction of the muscle cells, of the matured muscle cells or myofibers, but spares the PAX-positive satellite-like cells. And then what you observe that after another 3 weeks, roughly, there is quite substantial regrowth of muscle cells inside those tissues. And this is also paralleled by a contractile function, which you see here. So 48 hours after a cardiotoxin injury, these muscles are pretty much dead. They don't do anything anymore. And then if you wait, they regain a contractile function after 21 days. Not exactly to the degree they started with, but there is a functional regrowth. And this response is pretty much associated with gene changes that are pretty much similar to in vivo. So we have loss of more mature transcripts like titin. We have an early increase in cell cycle activity and markers that are basically indicate the muscle fusion. And also DHK1 was regulated in a way that compared to the non-injured control, we have a higher expression of DHK1. What about a heart muscle model? There's a couple of let's say, tissue-engineered models of injury models already published by different groups. We have also looked at a cryo-injury model where basically, and this is in brown, is all the cardiomyocytes inside this heart muscle ring. We place a cryoprobe and injure part of one arm of this ring. Here you see the injury site. This also causes a drop in contractile function, and this function also recovers typically over a time week of 4 weeks until we have a more or less complete recovery of contractile function. Again, also in this model, we see kind of gene changes that you wouldn't expect. We have a loss of myosin heavy chain 7, initially an increase in cell cycle markers, increase in some markers related to the extracellular matrix, and also DHK1, which increases at least not in the early phase, but in the regenerative phase later on. So how about the real heart and DHK1? So we looked at first of all, at heart biopsies from patients, from adult hearts, non-failing and failing. And there what we see is there's a clear downregulation of DHK1 expression in the disease state. And this is locally, basically, so this is heart muscle, but also if we look at the blood and serum, to look for the soluble DHK1, we find a similar thing, that compared to non-failing, patients with heart failure have a lower DHK1 abundance in the serum. In this case also there was actually a correlation to the ejection fraction, so the lower the ejection fraction, the lower the values for DHK1. So if we have the situation where DHK1 seems to be always upregulated in a regenerative state, but in heart failure patients, adult heart, it's actually down, we wondered if it would make sense to stimulate expression of DHK1 and to stimulate recovery potentially from heart failure. And to do that, we used a really nice model that was established in a lab by the group of Laura Celarayan, which uses CRISPR activation to be able to activate gene expression of any genes basically you want. So they generated an IPS, a stable IPS line with an enzymatic inactive Cas9 coupled to a transcription factor complex or activation complex, and thereby targeting those transcriptional activators to promoter regions of the gene of interest. And we use this line and this approach on cardiomyocytes, for example. So we derive cardiomyocytes from that line and then tested a number of different guide RNAs to see which one can activate expression of DHK1, and found one particular effective one that increased the expression about 20-fold compared to non-target control, which should not activate any particular gene. We also tested how these cells would perform in an EHM. So basically in this case we made engineered heart muscle with cardiomyocytes that were either transduced with a non-target guide RNA or with the DHK1 guide RNA E, so the most effective one. Both of them were combined with the same source of fibroblasts. In this case, these were primary cardiac fibroblasts. And then these EHMs were cultured in these kind of 48 well plates. And you can see that the tomato is reported for the DCAS in every cardiomyocytes, and the guide RNA transduction is monitored with eGFP expression, and we typically had a transduction rate of above 80%. So in basal contractile function, this did not seem to make a big difference. So both non-target and DHK activation cardiomyocytes formed similar performing EHMs. And we then asked if we apply an injury or a stress model, would we see any effect? In this case, we used a neurohumoral stress model, which means we treated the EHMs with a combination of noradrenaline and TGF-beta. And this 48-well platform allows us to basically non-destructively monitor EHM function over time without having to take out the EHMs. And this is what you see here. So this is day 29. So they have been matured for 29 days. And then this is the start of the treatment. And what you see is that you have an increase in beating frequency, which is obviously also the effect of the noradrenaline over time. And this is over 2 weeks in total. And you have a decrease in contraction amplitude resembling or as a measure of force of these tissues. So you see the decrease of force in the NAT-treated tissues. So how does DHK1 activation, specific activation in cardiomyocytes may affect this? First of all, we confirmed that this treatment gives us a similar response in terms of DHK1 expression compared to what we see in the adult heart. So the treatment reduces the expression of DHK1 in those EHMs. And using cardiomyocyte that with the guide RNA-E can really replace it or even increase it a little bit compared to the non-target control and the non-target tissues. So restores the expression of DHK1. We looked at the function after 1 week of treatment, so basically at this time point, when the tissues were treated for 1 week, and we did not really see any big difference at that time point. Yeah? So the force decrease of tissues was similar no matter whether they had a non-target guide RNA or the DHK1 activation. So in this acute model, we did not really see a difference, but we continue now to investigate this and look at the regeneration or let's say the recovery from such an injury and whether there's any difference in the case where you have DHK1 activation or not. So in summary, I hope I could show that you can model regenerative responses also in tissue-engineered muscles. In heart failure, local and systemic DHK1 expression is reduced, and you can activate using CRISPR-A approaches DHK1 expression, but in this acute model, it doesn't seem to preserve function. But I think in general, these models may help to dissect certain aspects of regeneration also in vitro. And for further discussion, there's a poster by Niklas. He's a medical student doing his thesis in my lab, and number 9. So if you're interested in DHK1 and like to discuss further, we would be happy to see you there. So thank you very much for listening.